Good morning, all. I am Akanksha Vishwakarma, Assistant Professor, Index Institute of Pharmacy, Malvanchal University, Indore, MP. I light the focus on this topic: cultivation, collection, processing, and storage of crude drugs. So let's start the lecture. Cultivation of crude drugs. Cultivation of medicinal plants requires intensive care and management. The conditions and durations of cultivation required vary depending on quality of medicinal plant material required. Advantage of cultivation: it ensures quality and purity of medicinal plants. Collection of fruit drugs from cultivated plants gives a varied and therapeutic quality. Cultivation ensures regular supply of fruit drug. The cultivation of medicinal plants and aromatic plants also leads to industrialization to a greater extent. Cultivation permits application of modern technological aspects such as mutation, polyploidy, and hybridization. And then we will see about the disadvantage of cultivation. The higher cost of cultivation drugs is compared to wild source. Loss due to ecological imbalance such as storms, earthquakes, food flood, drought, etc. We have finished about cultivation of fruit drugs, advantage and disadvantage of cultivation, and next point is the methods of propagations. There are the two main methods of propagation. First one is sexual method. It is also known seeds propagation methods, and the second method is asexual methods. It is also known vegetative propagation method. Uh, we will see about the asexual method, that is vegetative propagation methods. Vegetative propagation can be defined as regeneration or formation of new individual from any vegetative part of the plant body. The method of vegetative propagation involves separation of a part of plant body which develops into a new plant. There are two types of vegetative propagation. First one is natural method of vegetative propagation, and second artificial method of vegetative propagation. First, natural method of vegetative propagation. It is done by sowing various parts of plants in well-prepared soil. First, vegetative propagation by stem. There are the several examples are vegetative propagation by stem, bulb, squill and allium, corms, colchicum, tubers, potato and aconite, rhizomes, ginger and turmeric, shakars, pineapple and banana. This is the pictures of various types of vegetative propagation. The picture is showing root, bulb, rhizomes, etc. This is another methods of the vegetative propagation from root and stem. B. Artificial method of vegetative propagation. The method by which plantlets or seedlings are produced from vegetative part of the plant by using some technique or process is known as artificial method of vegetative propagation. Following methods are used: cutting, layering, grafting, micropropagation. First is cutting. There are the part of the plant such as stem, root or leaf, which if grown under suitable conditions develop a new plant. Stem cutting. Stem cutting are generally used to obtain a new plants. For example, sugar and rose. We can grow new plants by stem cutting the, from rose and sugar cane. Second method of vegetative and artificial propagation is layering. Roots are introduced on the stem while it is still attached to a parent plant. This part of stem is later detached from parent plant and grown into a new plant. Grafting. Third method of artificial propagation is grafting. So we will see about the grafting. A new variety is produced by joining parts of different plants. The rooted shoot of one plant called stalk is joined with a piece of a shoot of another plant known as season. For example, rose, citrus, and rubber, etc. We can grow a new plant by grafting. We can use rose, citrus, and rubber, etc. for grafting method. We have finished about grafting method and we will start about micropropagation. What is micropropagation methods? These methods consist of growing cell, tissue and organ in culture. A small piece of plant organ or tissue are grown in a container with a suitable nutrient medium under sterilized conditions. The tissue grows into a mass, undifferentiated cell called callus, which later differentiated into plantlets. And next point, they are then transferred into a pot or nursery beds and allowed to grow into full plants. This is the picture of micropropagation. First, we isolate the plant cells. Then grow into a suitable culture medium. The plant cells are converted into the callus, and the callus is uh, differentiated into plantlets. These plantlets are transferred into nursery beds or pots. This method is known micropropagation methods. So we have finished about the micropropagation method. This is another picture of micropropagation method. And uh, we have finished about the vegetative propagation methods. And then we will see about the sexual method. This is seed propagation method. First point is that in case of sexual reproduction, the plants are raised from seeds, and such plantlets are known as seedlings. For propagations, the seeds must be of good quality. We should select the good quality of seeds for propagation. Methods of sowing the seeds. These are the various methods for sowing the seeds. Numerous methods of sowing the seeds are in practice. Few of them are. First method is broadcasting. Broadcasting means throw from hands. Second method is dibbling. Dibbling means seeds dip into the soil. And then there are the many miscellaneous methods for sowing the seeds. We will see about broadcasting. In this methods, the seeds are scattered freely in well-prepared soil for cultivation. Second point, the seeds only needs raking. Third, if they are sown deep or covered by soil, necessarily thinning of the seedings is done by keeping a specific distance. For example, we can propagate the plants isabgol, linseed, and sesame by broadcasting methods in the field. This is the picture of broadcasting methods for the propagation of seeds. Next method for the seeds propagation is dibbling. So we will see about a dibbling. When the seeds of average size and weight are available. Second, they are sown by placing in holes. Uh, number of seeds to be put in holes vary from 3 to 5 depending on vitality. In the dibbling methods, we uh, place the seeds in holes in soil, and then we will supply the proper arrangement of water and allow to grow. This is the dibbling methods for propagation of seeds. So we have finished about the vegetative propagation methods and sexual propagation of methods, that is seed propagation methods. And we will see about the many miscellaneous methods or propagations of the plants. First, many a time the seeds are sown in nursery beds. Second point, the seedlings thus produced are transplanted into farm for further growth. Third point, such as cinchona, cardamom, and clove, etc. The picture is showing uh, nursery beds. Then we will see about the pest and pest control management. What is the pest and pest control management? First point, what is pest? A pest is an organism that cause epidermic disease associated with high mortality. Hence, pests are harmful to man physically and to his crop, causing economic loss. So, pest and pest control management is very important for the cultivation, collection, storage of the medicinal plants and other crops. So, it is a very much important step. We will see about some pest, type of pest. There are the different types of pest infecting medicinal plant are. First, fungi and virus. Second, insects. Insects means flying, moths, flies, and larvae. And crawling, beetles, beavers, and cockroaches. Third example of pest is rodents. For example of rodents is mice, field mice and house mice. Second example is rodents is rats, roof rats and Norway rats. Third types of pest is bird. There are the many types of birds. They can give the harm to crops and medicinal plants, pigeons, crows, starlings, etc. And other types of pest is other mammalians, such as snakes, cats, and dogs. So there are the many pests. They can give the harm to the crops and medicinal plants. So the management of pest is a very important steps for cultivation, collection, and storage of medicinal plants and other crops. Then we will see about the collection of the fruit drugs. First point is that collection is the most important step which comes after cultivation. Drugs are collected from wild and cultivated plant. Task for cultivation depend on collector, whether he is skilled or non-skilled labor. Drugs should be collected when they contain maximum amount of constituents. So collection of product is very important steps, and there are the many types of crops and herbs are collected from the different types. For example, tea. Tea leaves are collected when they are properly grew. And there are the different part of the herb are collected. For example, tea leaves are collected, Arjuna bark are collected as herb, and Ravolfia roots are collected as herb, and dried. The regions at which drug is collected is important as amount of active constituents change through the year. For example, rhubarb. It is collected only in summer because anthocyanin derivatives level is more in summer season and less in winter season. So season is very important when we collected the herb and crop. The leaves are generally collected just before flowering season. For example, Vashaka and digitalis leaf. Vashaka and digitalis leaves contain maximum constituents before the plants peel the flowers. So these leaves are collected before the plant bears the plant flowers. The flowers are collected before they expand fully. For example, clove and saffron flowers. Clove and saffron herbs are collected when the flowers are fully grown. Then, and the underground organs like tubers, roots are collected before plant cell die. For example, licorice and Rauwolfia. Some fruits are collected after their fully maturity, and some are collected when they are ripe. For example, the opium poppy are collected when they are raw, and tomato are collected when they are ripe. This is a picture showing the tubers of sweet potato. Sweet potato are collected when they are fully ripe and fully growth. And this is the picture of sunflower. Sunflower are collected when the flowers are expanded fully. And we will see some other examples of herbs. Baths are usually collected during spring season as they are easy to separate from the wood during this season. For example, Asoka bark, Cinchona bark, Kulchi bark. The picture is showing the bark is collected from the tree. as herb. The unorganized drug has to collect it as soon as they ooze out. There are the following many types of unorganized drugs used as herbs. For example, resin, latex, gums, etc. So we can say that the herb are collected when in proper season and proper time. So collection of the herb is very important, and the herbs and crops are collected as a proper methods is very important. So we have finished about the collection of the crude drugs, and we will see about another point, drying of the crude drugs. Drying of the crude drugs is very necessary step before marketing a crude drug. It is necessary to process it properly so as to first preserve it properly, second acquire better pharmaceutical agents. So drying is the very important step for collection, processing and storage of crude drug. Drying consists of removal of sufficient moisture so as to improve quality, make it resistant to microorganism, inhibit enzymatic reactions. Proper drying is a very important step. It reduces the microbial growth in crude drugs, and we can store the crude drugs very long time if the crude drugs are dried. And this is the picture. Uh, the crops are dry in sun. There are the many methods for drying the crops and herbs. And we will see about the methods of drying. First one is a natural method of drying, and the second method is artificial drying. Natural drying, we can dry the herb in sun. This is the natural methods of drying, and there are the artificial method of drying is the tray dryer, vacuum dryer, and spray dryer. We can use different types of dryer for the drying of the herbs. The last point is storage of crude drugs. We should store the crude drugs in properly and proper arrangement. First point, preservation of crude drugs needs sound knowledge of their physical and chemical properties. If drugs are improperly stored, quality can be affected. All drugs should be preserved in well-closed and airtight container. They should be stored in premises which are waterproof, fireproof, and rodent-proof. This is the picture of the crops are stored in proper manner. Some types of product are stored in airtight container, and some types of huts are stored in a bag. Thank you.